This is a paper model of a double helix, and that's origami. So when I proposed that we do this hologram, it was going to be a lot of steps. They went, no, Joe, like usual, you're climbing the logical ladder too quickly. We're going to do stereo images. But the publication was so successful that both my colleagues, postdocs, quickly got professorships, once now at USF and the other one's at University of Washington. And they actually are very much want to collaborate on this project. So we never did three-dimensional information in DNA. And so I kept it on the cooker. And I have a couple of friends. That's the Running Horse movie. That's Seth Shipman and Jeff Navala. Hologram, stereo images. And this is Eric Demain. Eric Demain is the youngest professor at MIT. He was homeschooled. He's at Computer Science and Artificial Intelligence, CSAO. And last year, working with a collaborator in Japan, they came up with an algorithm such that you scan any three-dimensional object, any complex three-dimensional object, and the algorithm produces the two-dimensional crease pattern for that object. So you can hold a lot of information in 3 dimensions. I'm also a consultant for SETI Institute out in California. And we're thinking that for next generation SETI, we should be interested in the genome. We should be interested in biological spacecraft, because nothing else can withstand the vacuum, temperature, radiation of the space environment. Is this one ready? No.
It's just unplugged, but otherwise it'll be ready.
Please plug. Oh, we're waiting for a dongle?
Probably.
Yeah, we're waiting for a dongle.
Do you have dongles?
No, it's coming.
Okay.
Hello. My beard is going to get in the way. Just clip it on my beard. So yeah, and there's all these nuances. We make all these mistakes in the search for extraterrestrial intelligence. It's, you have to reveal yourself to yourself before you can reveal yourself to anyone else. Aristotle knew that. He counted it as one of the principal elements of human tragedy, because we've never been able to do that. And so we send very serious, significant misrepresentations of Homo sapiens into space, however deeply encrypted. So, okay, hieroglyphs, cuneiform, Japanese kanji, these are all picture languages. And the fact that the literature, that the lexicon is written as pictures, must have some syntactical effects on the syntactical operation of the language, since those guys always have heads full of pictures. So here's the crease pattern for the DNA molecule. And using Eric Demain's algorithm, we could scan a complex DNA molecule with Van der Waals radii and such. But for now, first step, Mukund, myself, Yashas here, and the Art Science Bangalore, and now Seth Shipman and Czech Navala and George Church. Everybody's really excited. Everybody wants to collaborate. And somebody volunteered to do the DNA synthesis this morning, by the way. So yeah, you can make many complex objects with we can send more accurate pictures of ourselves than we've ever been transmitted. And the woolly mammoth. We're big on woolly mammoths at Church Lab. This is Nat Seaman. He was my colleague at Alexander Rich Lab many years ago. He's now a professor at NYU in New York. And in 1991, he won a prize for founding this field of DNA origami, which really has nothing to do with origami. There aren't any crease patterns, but he won his prize for making the first cube out of DNA with inverse repeats and cruciforms. So this is the icon, of course, of paper origami, the Japanese crane. And we're thinking to use NAD-Siemens protocol so that we would put the crease pattern from the Japanese crane, which is very simple, into this molecule that also contains all the cues, the inverse repeats, etc., for DNA origami, so that the molecule not only would hold the pattern, but it would fold into a simulacrum of the molecule, keeping a map of itself into itself. So I'm also affiliated with this guy's laboratory at MIT. This is Thomas Schwartz. That's one of my crystal radios hanging on the wall, by the way. And so I noticed that what we do is we make, like for instance, we made the first memory crystal. Come on, Klingons have memory crystals. Superman has memory crystals. Everybody has memory crystals in science fiction, but crystals are isometric, uniform repeats of the same thing over and over and over again, and you can't really modulate them to keep complex information. And so, but if the thing that repeats holds the information, and so that's what this is. It's a crystal of the microvenus, and we use synchrotron radiation remotely at a big national laboratory outside of Chicago to obtain the pattern, which looked like Rosalind Franklin's picture 51. We weren't satisfied. We wanted better data because actually there are few DNA molecules, double-stranded DNA molecules, relatively few have ever been crystallized, and there's still kind of mysteries, believe it or not, about the nuance of DNA structure. So when you beam of X-rays or synchrotron radiation through a crystal, you get this like diffraction pattern. It's like tea leaves, and if you're a crystallographer, you can actually look at these tea intuitively without Fourier processing, say that's a cone, or that's a cube, or that's a helix. But with sophisticated processing, you can tell intermolecular distances and interatomic distances and electromechanical bonds on each bond, electromechanical torsions on each bond. But I'm not interested in that. What I thought was, see, the DNA crease patterns, origami crease patterns are often radially symmetrical and in many cases highly resemble X-ray diffraction patterns. And so I asked Thomas if there is a way, if we instead interpreted this crease pattern as an X-ray diffraction pattern, could we work backwards and establish which geometric solid it was that refers to that? And he said, yes, it's possible. Now that in the Middle Ages and in the Renaissance, they thought that the Platonic solids had these mystical connections, and we often see pictures of the solid with fire in it or air in it or whatever. But now we can make a paper origami of fire, say, and then work backwards to geometric shape that has the fire in it. And this is a very elegant way to contain three-dimensional data. The relatively simple statistical description of the solid would contain, by this implication, a much more complicated structure, three-dimensional structure automatically. And so it's kind of magical, but Thomas Schwartz and George Church think it can be done. And so I have some crease patterns for horses, running horses. We're going to make the first running horse DNA movie, 3Ds. And how do you write a sentence? I mean, the idea for SETI and for deep time archives on Earth is how do we get around language? It's like mathematics is anthropic, much less language. And so the idea is how do you write a language into 3 dimensions? Like write a sentence like the elephant jumped over the fence. It presents an interesting problem. How do you make a map of the world that's in itself, that sits in itself? So then I thought about ways of writing mathematics and mathematical functions into this 3-dimensional world. And so there's number 1, a single surface. There's number 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 if you want to do base 10. But you can do, this is also 11, 12, and etc. There's an infinite number of geometric solids with an infinite number of surfaces. Anyway, so we're going to make the first three-dimensional information together as a collaboration here. I'm really excited about this. And Cocoon, like the other night, well, it was like 24 hours ago, something like that, we were like sitting, writing it all down on napkins, and he figured it out. The way to describe this origami pattern can be written in 46 mer DNA, or 48 mer DNA, which is unbelievable. A 96-bit line of code holds the data, and Mr. Michelangelo over here, Debbie's. Yeah, so we're all really excited. And my colleagues have already volunteered to please can we help, we want to be involved. We'll synthesize for you. DNA is on the way. Okay, now we can start the real show. Let's see.
That's fine. Thank
Can everybody hear me?
Yes.
Thank you for inviting me here. I'm really excited to be here. What a beautiful campus. I think every institution should have a mango plantation on their campus. So, a great transition came about when human beings gave up their hunter-gatherer lifestyle in favor of an agrarian one and invented many forms of agricultural and animal husbandry. The introduction and nurturing of unprecedented varieties of plants and animals accompanied this transition. It was a special power over nature that seems to have seriously impressed our forebears. Agriculture meant it was no longer necessary to make continual nomadic migrations in search of food. People produced surplus food and so had time to build cities and engage in trade with other groups. This surplus also meant that there was now enough time to develop specialized skills and find sufficient leisure time to produce art, science, and literature. Enough time to ponder the great philosophical questions of life, to construct mythology and religion that would help explain our place in the universe. And these legends were replete with myths about magical transformation of living material to become something else, something other, a kind of transanimation. Legends about Medusa, There are stories of golden eggs and golden fleece and golden apples and many more. Deities were given the prerogative to convert animate to inanimate and inanimate to animate matter. Daphne turns into a tree. I have to use the clicker. Do I have to use the clicker? I don't have to use a clicker, do I? Sphinx. She was a water nymph who was turned into water weeds while she was running away from this guy Pan, and she became Pan's flute. The Greeks were interested in perfect knowledge. They were obsessed with perfect knowledge of the human body. They noticed homologies in the geometries of... I hate this computer. They noticed homologies in the geometries of human physiology and other forms of nature. The art, architecture, and mathematics of the Greek Golden Age reflected biologically derived principles, and these in turn formed the groundwork for the arts and sciences of the European Renaissance. So one of my favorite stories is transanimation stories, is about the sculptor king Pygmalion who carved this beautiful woman and then it was so beautiful that he fell in love with this chunk of stone, and he was weeping, and the goddess felt sorry for him and brought the sculpture to life. That was Galatia. Yes, Greeks and architecture. This is Michelangelo's Moses, and in a moment of frustration, there's this folk tale that he said to have hurled his sculptor's hammer at the marble sculpture, saying, "Perché non mi parli? Why don't you speak to me?" He wanted his sculptures to come to life. This kind of thing repeats itself throughout the history of art and literature. Oscar Wilde's picture of Dorian Gray, where a painting animates itself with the evil deeds of its subject. Robert Louis Stevenson and Dr. Jekyll and Mr. Hyde, same sort of scenario, a kind of transanimation. The myth and folklore of our own age resonate with these same ideas. Power over the secrets of life are now relegated to monsters and evil guys, however. But artistic treatments of the human form still call for knowledge of human anatomy and physiology. It's a legacy that we still carry on today. Artistic treatments of the human form, yes, still call for. I'm jumping ahead, I think. It vastly increased understandings of molecular medicine and human genetics with cybernetic prosthetics, artificial organs, and even artificial blood. But now, transanimation is a technological reality. We have grown rat, mouse, pancreas, and human mouse livers made from human cells, and even the human mouse lung. But in my opinion, perhaps the most profound example of this transanimation can be found in the search for extraterrestrial intelligence, because by simply asking a question, we're attempting to bring the whole universe to life. And for many years, many aspects of my work have been either directly or indirectly connected to the flaws and fallacies and crazy philosophical questions involved with the search for extraterrestrial intelligence. I've carried out 2 radar transmissions across interstellar distances to sun-like stars. These are the most powerful and the most lengthily studied transmissions ever undertaken. Poetic Vaginale in 1986, 2 million watts from Millstone radar at MIT Haystack Observatory. And in 2009, the Rubisco Stars transmission from Arecibo Observatory in Puerto Rico. I carried this one out on the 35th anniversary of Frank Drake's famous SETI transmission, Search for Extraterrestrial Message to Extraterrestrials. The Rubisco message contained data precisely corresponding with the gene coding for the most abundant and one of the most ancient proteins on Earth, 1,5-bisphosphate Rubisco. Ribulose 1,5-carboxylase oxygenase. It's not only the most abundant and one of the most ancient, but it's among the least efficient proteins, which helps to account for its relative abundance. But it tells a great story about life on Earth. It takes inorganic carbon from atmosphere and turns it into food. But anyway, long story short, there are 3 basic problems with all messages for extraterrestrial intelligence. And one is you have to have something that can persist for periods of geologic time, because in a cosmic circumstance, even the speed of light is a slow boat. You have to have something that can withstand the extremes of the space environment, radiation, temperature, vacuum, and you have to be able to create conveniently hundreds of billions of copies for hundreds of billions of possible receivers. And for this, bacteria, especially sporulating bacteria, answered all of these problems very well. And this is what brought me to molecular biology many years ago. I created my first message-bearing bacteria in the 1980s. This was Microvenus. It was an icon coded into DNA using a strategy that was in part one of the mathematical strategies used to code radar signals for extraterrestrial intelligence. I'm conscious of the matter of time here, so I'm not going to explain all this too deeply, but it was coded into DNA by giving phase values to DNA bases, which in turn coded for a binary raster that holds the Microvenus icon. This is the Microvenus sequencing gel. In it, In addition to its poetic motivations, Microvenus was created to present a possible technical solution for all the problems associated with all attempts to use spacecraft and radar for transmissions for extraterrestrial communications. Microvenus explored the notion that biology could play an important role in sending astronomical numbers of directed messages across many thousands of light years and correspondingly enormous periods of time. Microvenus was the first genetically engineered work of art and the first use of bacteria to model biological carriers of interstellar messages. This was the DNA supercode, the first of several programming languages created over the next several decades to code text and images into the DNA of living organisms. The first high-resolution digital image encoded into DNA was the Milky Way DNA, encoding an image captured by NASA's Cosmic Background Explorer, the COBE satellite, into the physical minutiae of DNA molecules. The Milky Way DNA juxtaposed these vast differences in scale. COBE images were the first images human beings were able to capture of the galactic core of our own galaxy, and the first maps ever to be constructed of the galaxy itself. So again, I can't go into details of the operation of this code at the time, but I invented the supercode to make something that could keep parity with input data, that could compress information, but that also would be biochemically and biologically friendly with respect to the host organism. So I've done many projects about what we would call this transanimation idea. And the other night at the British Council, I met a young woman who's into fabric, and so I threw these slides in case she's here. I worked with Hideki Suzuki a few years ago at the Transgenic Silkworm Research Laboratory of the National Institute of Agrobiological Sciences in Tsukuba, Japan. We modified silk moths with genes from this marine sponge, Tethia rancia. We gave the silk moths a gene called silicatine that polymerizes silica in its natural use. Polymerized silica from silicic acid, the most abundant soluble form of silica in seawater. And in the sponge, silicatine forms these filaments which create the spicules, the silica glass spicules, which are the endoskeleton of the sponge. But see, okay, so, oops. Yeah. Spicules, silicic acid. So silicatine is a promiscuous protein, and if you starve media for silica and instead give it another heavy metal, but silicatine it tends to chelate that metal. And we used bacuvirus-based transposase expression vector to create a gene fusion with fibrin, the principal silk protein, and silicatine. And then it took 2 years to establish a stable strain, on the way which I made these glowing green silkworms, which the art world would have loved. Oh, you made green worms, but they always make green worms. But what I am proud of are these silk fibers that are biomineralized with metallic gold, where gold is, in electron microscopy, gold is integrated into the silk structure. Okay. So here I would have us return to another one of the historical episodes where living material magically transforms into something else. Genesis 19 in the Hebrew Bible tells us a story about the patriarch Lot, whose wife is turned into a pillar of salt. I have a special connection with the story because I found myself bringing creatures locked away in ancient salts for hundreds of millions of years back to life. In fact, clues to unlock some of the most profound scientific mysteries about the history and origins of life just might be found in crystals of ordinary salt. So this story starts here. These are my, I build these weird analog instruments sometimes in lab for different purposes. I built a fishing microscope that you can hook a fishing rod up to, and you can struggle with the paramecium and force feedback auto ramped up to feel like a 300-pound tuna fish. Actually, there was a haptic research group at Media Lab, so like where people who are blind, who can never peer through a microscope, could actually feel the paramecium and the stentor. They feel different. Like when you're fishing, you know, a trout feels different than a catfish, and they loved it. So I had an exhibition. I also made an audio microscope that you can listen to. Again, I have files, but really we don't have time. So I had an exhibition in Nantes, France, at La Unique, and as a result, I was running around to lakes and ponds and rivers in the region to collect microorganisms to listen to. And my friends from Paris came and said, Joe, we're going to bring you to the salt pans, medieval salt pans, in Garonne, Brittany. I arrived, and there are these salt pans said to be created by the Vikings 1,000 years ago, where they make the famous and very tasty blue Celtic French salt. And I sampled the organisms in these salt pans and was blown away. They were just hugely abundant, wildly weird. I never saw anything like it in just diverse stuff swimming around. And I wondered, well, what the hell happens to these? These are halophiles. They're diverse and extremely diverse salt-loving organisms. I did a little literature search, and I found that some food scientists had investigated salts as possible carriers of pathogens for rubbing on cheeses, and another group in France tried to create these biological fingerprints for salts from different regions to avoid counterfeiting each other. But neither one of these studies externally sterilized the salt, so they're getting contemporary organisms with what other organisms might be in the salts. So I went to the food store, the fancy food store, and I bought all these boutique salts from all over the world, and then I used ethylene oxide to externally sterilize the surfaces of the salts, and then I created media. That's ethylene oxide sterilizer, ethylene oxide indicator, and I made media, several forms of hydrosaline media, inoculated, incubated, and lo and behold, I got growth from these various salts. But one of the salts I got growth from was not sea salt, it was mineral salt. It was the Himalayan salts from Pakistan. And these salts are 250 million years old. And that blew my mind. They say the Himalayan salt is pink because it has iron in it. These are the organisms you can find growing in Himalayan salts. So let's see, these are halophiles from Djibouti, Lake Bissau. Some of these are movies here. This one. So I found all these communities of organisms embedded in these salts. And I thought, I got a fisherwoman friend of mine to go outside to Boston Harbor to get me some clean seawater, which I put through a coffee filter, and then I autoclaved it, and then I inoculated it with the French salt organisms. They grew just fine. Watch out, your salt could be counterfeit. These are the organisms in the Pakistani salts. These, yeah, well. So, the legendary philosopher's stone was not only dear to alchemists and sorcerers because it could be used to transmute base metals into gold, but also because it was reputed to hold the secrets of everlasting life. And while many scientists worldwide, including several church lab members, are conducting research on aging, no one has yet been able to produce the 5-year-old mouse. So now, suddenly, we have mineral salt crystals that can apparently keep things alive for 250 million years. At this point, I organized this great Paleozoic salt expedition in order to acquire all the mineral salt samples from all over the world. And in case you're wondering, it's really dark down there. This is a search for ancient salts that geologists call primary salts, salts that have never been redissolved and recrystallized since their initial deposition. They're found deep underground where they're protected from climbing and weathering. This is Berchtesgaden in Germany, Hallstatt, Austria. I also obtained salts from Heilbronn in Germany associated with the Permian-Triassic Muschelkalk in northwest Switzerland, 235 million years old, 200 in Upper Triassic. Also 225 million year old salts from Bex, and 25 to 20 million year old Miocene salts from Armenia. I'm also expecting new samples from Thuringen, Germany, and somebody yesterday or this morning promised to introduce me to Indian geologists who may find some good samples here. I also went down into the deepest salt mine in North America at 2,300 feet on Lake Cayuga in New York. And this is my philosopher's stone, approximately 485 million years old, and I have this big collection in the laboratory of all the salts that I've collected all over the place. So what's going on here? Salt crystals expand and contract at a different rate from the brine in which they're situated in, and as a result, they include these tiny little inclusions of fluid and gas. They're called fluid inclusions, and this happens in all salts. And some genius, and I don't know who it was, I've been trying to find out, there's an analytical technique in geology called fluid inclusion microthermometry. And what they do is they find salt with one of these fluid inclusions, and all salt gets these fluid inclusions wrapped more frequently at the center of the crystal. And put it on a microscope, refrigerated microscope slide, and then cool it down until contraction causes a vapor bubble to form in this vacuole, in this inclusion. And then they slowly heat it up until the bubble disappears, and that's the temperature at which the crystal formed. It's a window on ancient climatology and paleoceanography and good, fascinating method analysis. But then in 1960 and in 1963, two biologists, two groups, said they had found organisms in these fluid inclusions in ancient salts. But this was the era before molecular biology, and so there was a lot of skepticism about these reports, pointing to the possibility of external contamination. But since that time, survival of microorganisms over periods of geologic time is difficult to imagine, and contamination issues were suspected. Since the late 1990s that picture's changed. Later findings found that prokaryotes cultured from ancient halides, Permian salado salts from New Mexico, were full of halophilic archaea and a halotolerant bacterium, Virgibacillus. Atacama desert deposits yielded Permian age organisms from 2,000 meters in depth, etc. There were some other studies. These are some of the organisms found. And one of the recent findings, which was amazing, because archaea were never known to make spores, but Halobacterium salinarum, one of the most studied archaea, create these spore-like structures. They've been found in cyanobacteria and Dunaliella, an algae. Have been found in gypsum, another evaporoid, in quarries in Italy. And so bacteria, algae, and archaea have been found in gypsum and calcite evaporites, as well as in salt. Earlier protocols in decontamination techniques consisted of immersion in sodium hydroxide and hydrochloric acid solutions, which of course also dissolve salt. Usually, and hyposaline media. Then the early work was done with microdrilling, which is an invitation to deliver external organisms from the outside the surfaces to the inner surfaces. So I checked on my ethylene oxide sterilization with GFP, and since then I've done more elaborate controls, incubated my ancient salts, and lo and behold, from all over the world and across time, this one is not a movie. This one is not a movie. These are 485, let's see, the one on the right is a 485 million year old. And this one is a movie. Well, anyway, we got DNA. These are 16S studies, the control, and these are the organisms. Green is the most likely, but the reason the species are unknown, so they're not noted, and less likely are more contemporary species noted in the browner colors. So this one is, on the left, is 250 million year old salts from Pakistan. The right two. Here on the right are 438 to 419 million year old salts. And more 250 million year old salts. So we were like worried, does ethylene oxide really work? And so we ethylene oxided, and then we systematically flame sterilized for 30, 60, 120 seconds and did as much handling in sterile lids as possible. And the salt got so hot, it must be a great thermal insulator, because it got so hot that when we dropped it in media, it's horrible media. But we got DNA, we got organisms, and we got DNA. So, it might be, it turns out, there might be good reasons to extend this search into space as well as time. These several meteors have fallen that have been found to contain salts with fluid inclusions. Among them is the Monahans meteorite that fell in Texas. These are fluid inclusions found in that meteorite. And these were studied by an acquaintance of mine at Johnson Space Flight Center, Mike Zielinski, who surveyed these meteorites, this one and the Zag meteorite, which is the second salt-bearing meteorite found on Earth, for the presence of iodine-129, an isotope of iodine. And with this survey, he was able to, this analysis was able to determine that these meteorites are 4.5 to 4.7 billion years old, which is older than Earth. Both are thought to have originated in this asteroid that's in a kind of harmonic orbit with Earth's synchronous orbit of Earth, the 6 Edie, which is ironic if she's so old because Edie is the goddess of youth. They think that 40 to 70% of all meteorites that enter the Earth's atmosphere come from this asteroid. So these are several samples that are found on the market, 138.9 gram sample of the Zag, 1.4 kilogram sample of the Zag, and an 18.8 kilogram largest intact Zag meteorite mass in the world, bargain at $50,000. So it just so happened that about 2 years ago, I was invited to present for a convention of venture capitalists at Oxford University. And I mean, there were billions of people in the room, and I told them that they shouldn't wait for the money and that they should just go ahead and directly save the world, and what you'd expect me to tell them. But I learned something very interesting. I learned that there are two species of venture capitalists, one of which is a manager for funds for many people, for large organizations, many people, shareholders and such. And the other is the manager of funds for a family. And the latter is more likely to act on principle and conviction than the former. And so there is a brisk meteorite market for all these meteorites, and the more famous the meteorite, the more valuable it is. So I propose that I would, of course, take some of these meteorites for analysis, but that the surviving fragments could be put back onto the market. They could get all or most of their money back. But that if I found what I thought I might find, then, of course, the value of these materials would increase exponentially, and it's the kind of language that venture capitalists like to hear. So, after several months of MTAs and bizarre meetings with lawyers, the meteorites arrived in my laboratory. That was exciting. They're still exciting. Then here we are, like Michelangelo at the laboratory bench, chipping stone with hammers and chisels, looking for indications of salt, incubating. I have liquid-culture and agar, but now we're doing anaerobic cultures as well, and we're finding these things, and I don't want to like run around and go, aliens, aliens! In fact, George Church and I are talking about when we publish, we'll just say, okay, new organism described, we'll tell everything we know about this organism. The very last paragraph will go, and it was found in this meteorite. This is a movie of something I found swimming around. And I found stuff swimming around in different directions to rule out actually had better movies to rule out diffusion. This is the DNA we found by a nanodrop. We didn't find DNA by a qubit. I did propylium iodine staining, which I got really excited about. Look at all that DNA, but then I found out that the meteorite autofluoresces red. So much for that. So halophiles don't always, even terrestrial halophiles don't always work with the standard tools of molecular biology. Universal primers aren't universal. You don't use TAC. There's a lot of things we're working out, whole new protocols and different molecular operators now. I'm doing anaerobic cultures. I finally obtained anaerobic growth chambers. And I found some really interesting things. Like years ago, I grew magnetotactic bacteria, and I found that that's a magnet in my, that's a magnet. And that the sediment that appears in these cultures is highly magnetic. It's almost like a magnetic fluid. And then, so I decided I would, after my experience with magnetospirillum, I decided to take magnets on culture flasks. Here on the right is on your right is a little booger in the copper tube. That's at one of the magnetic poles of the magnet that's taped on there, and that's magnetospirillum. But when I did that to my to my meteorite cultures, I got stuff that also had one magnetic pole and not both magnetic poles, and this weird red biofilm, which appears underneath some different cultures too. I have more slides, but didn't have time to integrate them this morning. The magnetic bacteria, they make these little organelles called magnetosomes, in which they sequester perfect crystals of, in one case, the most studied one, magnetospirillum, is magnetite. The next most studied one contains little crystals of a paramagnetic material, iron pyrites. But what none of the papers mention is that all of the minerals that they find in these magnetosomes are semiconductors. They're the same crystals I would use to build crystal radio sets. And I thought, wow, what's going on here? And so far there's no real reasonable explanation of why we have magnetosomes in the first place, why bacteria are magnetotactic. And recently it's been shown that there are magnetotactic protists as well. So the first explanation is, they follow the field lines to get their food, but in Massachusetts the field is 72 degrees, closer to the equator, it's more horizontal. At the poles it's vertical. And I had frequent mutants in my growth of magnetospirillum where they went to both poles, and so that question is sort of ruled out. But all of the magnetotactic bacteria are helical, and there are two proteins, MAMJ and MAMK, that line up the magnetosomes in the center of the cell, so they follow this helix. And you don't have to have a coil to have inductance, but if you have a coil, you have back inductance. So a resonant circuit, you need capacitance, you need a semiconductor, and an inductor. And so bacteria have a difference of pH from the outside to the inside, which allows for cell membrane transport and such, but that also equals the difference in capacitance. And the inductor is the line of magnetosomes and the little crystals in the magnetosomes of semiconductors. And so I wondered what, everything, all of these have characteristic frequencies. Capacitance has frequency depending on its charge-discharge mechanism organization, and the inductor has a frequency based on the radius of the turn and the thickness of the wire. And so I went back to all the Faraday's, for all the rules of electromagnetism, and I calculated with MATLAB the resonant frequency of these bacteria, and it turns out that it's cosmic microwave background, the frequency that transmits to atmosphere, the best. It comes from every direction in the universe, and it's redshifted over time, of course, but so these things have evolved. So I'm just saying that's really interesting. Okay, so I'm almost finished. I'm 2 minutes over, and I'll be finished in 1 minute. Venus of Willendorf, she's the first page of every art history book. She's in an 11.1 centimeter high, 4.4 inch Venus figurine in Vienna, now in the Natural History Museum in Vienna, and she's estimated to have been made between about 28,000 and 25,000 BCE. But in terms of human genetics, Venus of Willendorf is only a youngster. The mitochondrial Eve is the most recent, said to be the most recent common matrilinear ancestor. A mitochondrial, she's thought to have lived later than Homo heidelbergensis and the emergence of Homo neanderthalensis, but earlier than the out of Africa migration, roughly 70,000 to 135,000 years ago. A 2013 study cites a range of 99 to 148,000 years for the age of mitochondrial Eve. She's about 3 or 4 times older than Venus of Willendorf. Of course, there's an older Venus still. One theory is that she spontaneously appeared sometime in the Hadean age on a dramatically different Earth than the one we know today. Earth is thought to have formed about 4.5 billion years ago in a primordial swarm of debris left over from the formation of the sun itself. For hundreds of millions of years, it was very probably mostly molten and heavily bombarded by other materials lingering in the ancient protoplanetary nebula, and sometime early in its history, the Earth is thought to have endured a cataclysmic event of astronomical proportions that resulted in the formation of the Moon. The currently favored hypothesis for the formation of the Moon suggests it was formed out of debris left over from a collision between Earth and an astronomical body the size of Mars. During the course of its formation, Earth is thought to have experienced dozens of collisions with such planet-sized bodies, but the moon-forming collision would have been only one such giant impact, and certainly the most significant impact or event. The late heavy bombardment occurred later, approximately 3.9 billion years ago, which is the age of these critters, recently confirmed by NASA's SIMS analysis, secondary ion mass spectrometry, that these microfossils date to 3.5 to 3.4 billion years, and so may very well have overlapped with the heavy bombardment. And what's really amazing is these biological pioneers were highly refined from a systems perspective, already capable of photosynthesis. It's like, life made its early appearances with plenty of relatively sophisticated accessories. They had two-car garage, and an air conditioner, and a refrigerator, and how in the hell? So now we're designing protocols to search for indications of an extremely ancient, even a few collected remnants of the primordial solar protoplanetary nebula. So this is like this word panspermia. It's like in light of recent events, we need to find a less male-centered term, I think. And I'm open to suggestions. Panzygodia, any ideas? I'm open for new ideas. Thank you very much, ladies and gentlemen.
Maybe we'll take 3 or 4 questions. Yes?
Joe, could you elaborate on the cognitive background radiation hypothesis?
It would be,
Premature?
I don't know. I think it clearly there's it could whether there is some other purpose for magnetosomes and magnetotactic characteristic bacteria, that it's also probably can be used as a proton or electron source, depending on which end you're looking at. And I mean, as a source of energy, it kind of makes sense. We find all these organisms deep in your subsurface, which can be penetrated by cosmic rays. Cosmic rays are way cool. Cosmic rays and cosmic microwave background. No, I intend to, like, George wants me to publish the stuff about the resonance of the terrestrial magnetobacteria. I just thought that was really interesting. I never thought it was publishable, but he went, Joe, you've got to publish that stuff. So I want to do kind of parallel experiments with the materials that I find, and I think I have magnetotactic stuff growing in my cultures now. And one really interesting thing is that, heat usually deactivates a magnetic field. But the stuff in my 45-degree incubator is highly magnetic, like ferromagnetic fluids. But when I sit it on the bench for 4 or 5 days and it cools down to room temperature for a while, all of its magnetic properties seem to disappear. So I don't know what the hell is going on.
I'm sorry, this wasn't very apparent to me, but what was the rationale you used? I mean, what is the rationale when you need to choose media for sample from meteorite?
The rationale? Oh, because they were found with salts with fluid inclusions. The only meteorites ever found with salts with fluid inclusions. So what was the rationale for me to go after the meteorites in the first place?
Liquid cultures?
Yeah. Liquid cultures and agar clays. And now anaerobic cultures, which makes sense. I haven't done anaerobic cultures yet. I should have done it first.
One question. So apart from sequencing 16S, you could sequence all the DNA they have. You probably have done it.
Well, see, it's the molecular operator's problem. It's like, can I build libraries from this stuff I see swimming around? But it's like, how do I build a library if my reagents don't work? So we're going. So I get variable results. Sometimes it works, sometimes it doesn't. Now these experiments are being repeated in Israel and in Sweden by 2 collaborating laboratories. I mean, I'm trying to be as careful as possible.
Okay, great. Thank you, Joe.